How training your brain affects neuroplasticity and makes you smarter. My name is Leanne Poston. My background is a trained pediatrician and physiology instructor, and I'm here to discuss how training your brain affects neuroplasticity and makes you smarter. By the end of the session, you should be able to describe and draw the structure of a neuron, explain how neural circuits can be changed, discuss how training affects neuroplasticity, define intelligence, list factors that affect intelligence, summarize the relationship between a growth mindset, deliberate practice, and intelligence. As a medical student, you have so much to learn and so little time to do it. You will be balancing trying to read 2 or 3 textbooks at a time, researching, and then trying to review past content to prepare for step 1. You have strategies for studying in college. They may or may not work for you. Medical school is also expensive. A failed course requires remediation or repeating the course. Multiple failed courses require repeating the year. This is a huge expense. Are you sure your current strategies will work for you? In the past, you have always relied on evidence-based practices. You have been taught to evaluate the data, look carefully at the procedure, and make sure that the experiment could successfully be replicated. In this presentation, we will start with concepts you are familiar with, the components of the central nervous system, and describe an evidence-based method of study that may not feel like it makes much sense at first, but it is backed by research and has been replicated by many educational scientists. First, we will explore the neuron, or the functional unit of the brain. Then we will look at how action potentials cause the release of neurotransmitter, which cross synapses and provide a binary code for transmission of information. These neurons are organized into neural networks. All components of this process can be modified by repetition and goal-directed training. The process of change is called neuroplasticity. The neuron is the structural and functional unit of the nervous system. We are born with 100 billion nerve cells. There are 4 main components of the neuron: the cell body, the axon, the dendrites, and the synapse. The cell body contains the nucleus, which houses the genetic material for the cell called the DNA. The cytoplasm of the cell body contains the organelles of the cell. The dendrites are the receptive area of the neuron and receive messages from other cells of the brain. The axon is the part of the neuron that transmits messages to the next neuron. The location where one axon comes in contact with the cell body or the dendrite of another neuron is called a synapse. Chemical messengers called neurotransmitters carry the message from one neuron to the next. The production of these neurotransmitters make the neuron one of the most metabolically active cells in our body, using approximately 20% of our resting metabolic rate. The axon at rest has a slightly negative charge on the inside of its membrane compared to the outside. This state is called polarized. To send a message, the axonal membrane is depolarized. This is accomplished by successive opening of sodium and then potassium gates. You may recall that both sodium and potassium are positively charged ions. If you open a sodium gate, the inside of the axonal membrane becomes temporarily positive, and then it is restored to its baseline negative state by opening potassium gates. This generates an electrical message called an action potential. The message is generated at the beginning of the axon in an area called the axon hillock. The action potential, or electrical depolarization, is transmitted down the axon to the synapse. It is an all-or-nothing potential, which means that there is no degradation of the electrical potential from one end of the axon to the other. Just like the electrical wire, the neuron has an insulator which retains the charge inside the axon. This insulator is called a myelin sheath and is a fatty cell surrounding the axon. Where one neuron comes in contact with another, it is called a synapse. In simplest terms, you can think of a synaptic connection as a communication in binary code. When you send an action potential, it is on. and when you do not, it is off. You can have summation of these on and off signals from the thousands of neurons that synapse on a single neuron and have an infinite number of possibilities, just as the binary code used by a computer has an infinite number of combinations. The brain makes use of this code to provide the information needed to account for every thought, feeling, or action that we take. The number of synapses peak at the age of 1 to 2. This number will decline by about 50% before we reach adulthood. In adulthood, we have approximately 150 trillion connections left. Why the decline? It could be that neuronal synapses that are not used are lost, and it also could be due to genetically driven pruning of the synapses for maximal efficiency. Ironically, during the time of maximal learning, there is little change in the number of synapses. Research indicates that the overall structure of the brain is genetically determined, and the neural networks are shaped and pruned by experience. The more neural connections that we have, the faster our mental processing may be. As mentioned earlier, myelin coats the axon and allows the action potential depolarization to be transmitted from one end of the axon to the other. The thicker this myelin sheath, the greater its insulator potential and the faster the transmission. How can this myelin sheath be changed? The thickness of the myelin sheath is correlated with ability. Increased practice and training can increase the thickness of the myelin sheath and the corresponding neural pathway. The increased thickness of myelin acts as a better insulator, which results in increased strength and speed of electrical signals. The more we send action potentials down the same axons, have them cross the same synapses, and make connections in a predictable way with the next group of neurons, the faster the entire process becomes. The brain chunks these combinations of signals and neural pathways as cognitive motor activities and stores them. Neural networks that we use when we consciously do something are not the same ones as the ones we use when an action has become automatic. The basal ganglia, which are gray matter structures deep in the brain, direct these automatic actions. Extended training and repetition can lead to chunks of information being encoded as automatic or reflexive. This chunking of information with proper training can lead to the ability to allow actions to occur so fast that conscious processing cannot keep up with them. Starting with ICM, you learn how to interview, perform a physical exam, and then how to develop chunks of diagnostic information. You also learn to identify when there's a life-threatening emergency and when you have time to pause and consider a greater array of diagnostic possibilities. A growth mindset focuses on the doing instead of the results. If you understand neuroanatomy and physiology and can see that repetitively sending action potentials down the same axon will result in thickening of the myelin sheath, an increase in the number of sodium and potassium channels, and an increase in the amount of neurotransmitter released per action potential, then you can understand how training can make you better at learning something. In addition to the changes in an individual axon, there are the resulting pruning of synapses and changes in neural circuits that make the pathway more efficient. So how does this apply to medical school when you don't have time to go over every fact until it becomes reflexive? This is the part you might not like. You have to have some desirable difficulties. It is so much easier to read and reread material until it feels familiar. But this is passive learning. In order to get the greatest amount of change in the synapses in the shortest period of time, you need to actively retrieve or recall information. The harder this process is for you, the more physiologic changes you are making. If students set learning goals for themselves and try to reach these goals with focused training, they will fail many times, but they're not afraid to try. Failing to meet a learning goal does not mean they are not as intelligent as they thought. It just means one area of training requires more work. Each setback shows you where you need to focus your efforts and your performance continues to improve. For this reason, Carol Dweck suggests that we should praise the efforts students make towards their goals and not their success or failure. Deliberate practice is goal-directed practice. where you continuously try to extend your performance boundaries. Deliberate practice is usually not enjoyable. It is hard work. It has been demonstrated that people who have a hard time identifying their shortfalls when they are learning something new, so effective training usually requires a coach or a trainer to help identify these shortcomings. Anders Ericsson says that the average person spends 10,000 hours or 10 years of practice to become an expert in their field. Understanding this may help you understand why paying for someone to lecture to you is not a good use of resources. You need trainers or people who can identify your strengths and weaknesses and your diagnostic skills. Boonshop School of Medicine has responded to this research by developing a curriculum that minimizes lecture and maximizes training and feedback. So knowing that anyone could be an expert if they invest enough effort and deliberate practice in a goal-directed way, then what is intelligence? There are two types of intelligence: fluid intelligence and crystalline intelligence. Fluid intelligence is processing power or short-term memory. Crystalline intelligence is long-term memory, or the range and extent that information is stored and able to be recalled. There are also two main contributors to level of intelligence: genetic and environmental. Environmental factors that affect intelligence potential include socioeconomic status, nutrition, and early exposure to educational stimuli. The genetic contributors to intelligence are the maternal and paternal genome. Even this is not as straightforward as you may think. There is a series of nucleotides that make up the genome and provide the genetic code, but there is also the epigenome. Viruses found in our genome affect the genetic code, as do mutations and errors in replicating the code from cell division to cell division. Hormones also affect the genetic code. As an interesting example, environmental conditions that a grandmother lived under can change her hormonal levels, which can affect the genome of her future son or daughter that is in her womb during these hormonal changes, which can then affect her grandchildren. This was most completely studied when looking at generations that lived through and after the Dutch hunger famine. The starvation of the grandmother led to changes in her offspring's genome, which led to changes in the grandchild's genome, causing them to hold on to calories to prevent starvation. This has been demonstrated to be a cause of obesity that is not due to overeating. If there are so many factors that affect intelligence, and there is so much to learn about both the genetic and environmental contributions to IQ, then we should focus on the factors we can change. There is so much accumulating evidence that training can change both the anatomy and the physiology of the brain. Focus on the skills you want to improve, develop learning goals instead of performance goals, use deliberate practice, commit to the fact that it will be a long-term effort, and be prepared to be successful. If you are tasked with learning all the muscles in the body, sit down and divide the task into learning objectives. Develop a time schedule for the learning. Make sure that you space out your study sessions. Cramming doesn't work. It only uses short-term memory. There is no encoding and consolidation of information, and therefore no long-term, durable changes to the neural networks. Use diagramming, concept maps, models, and written descriptions to work on each section that you need to learn. After studying a single section of the body, go back to a previous section and quiz yourself, or have your lab partner quiz you. Metacognition is the process of monitoring our own thinking and looking for errors. Try to generate questions that you will think that you will be asked. Formulate an answer, and then check your work. This will help you identify any errors in your thinking. Interleave the study of physiology of these muscles in with the study of their anatomy to make it easier to come up with cues to help you recall their names. After you learn all the muscles, continue to go back and retrieve their names after you move on to other units. Testing is a learning event. Each time you or your instructor tests you, they are forcing you to retrieve information and helping you identify the weak areas of your knowledge. In summary, set learning goals, not performance goals. Use deliberate practice instead of rereading and other passive methods of repetition. Interleave the study of multiple topics and then tie them together with concept maps or diagrams. Finally, Know that your hard work is changing the structure and function of neurons in the brain so that each time you work hard to retrieve the information, the neural connection transmits the information faster and the connection becomes more durable. No longer do you need to worry about whether you are as smart as your fellow classmate. You are in charge of your own learning. Your trainers stand by ready to help you. Growth will only occur if you use the active learning process as described, and if you embrace feedback, knowing it will be difficult to identify your own weaknesses, and you rely on trained clinicians to help you with this. Someday, your patients will thank you.